So, hello. So first of all, I would just like to thank Sofian for organizing these ICMS meetings here. And I would also like to thank for the opportunity to giving this talk, even though it was with a fairly short notice. But today I will present about a very new project in our lab that has been a characterization of illegal compounds on the GABA-A receptor. So I work at Linköping University, where I'm responsible for our electrophysiology infrastructure. And this is connected to the National Chemical Biology Consortium, Sweden. And let's see if it's on. Yeah, as you can see here. So different projects from across Sweden can apply to us to get our assistance to develop the assays, but also then to do recordings on our QPatch system. So there are quite many electrophysiologists in Linköping, but the three of us that are involved in the CBCS team, that is Professor Fredrik Linder, and it's me, and it's also Amaya as a molecular biologist. So we can help users both with voltage and ligand-gated ion channels. And some of you may know that my expertise is not within ligand-gated, but I've been working with voltage-gated ion channels, in particular voltage-gated potassium channels for quite many years. So this project came to us from forensic scientists. So Linköping is also the Center for Forensic Scientists in Sweden. So this project came to us from Professor Henrik Gren and postdoc Caitlin Norman. And they are also connected to RMV, that is an abbreviation for Rättsmedicinalverket in Swedish. So that stands for the National Board of Forensic Medicine. And the use of electrophysiology have not been really high within the field of forensic medicine before, in Sweden at least. But they are very happy now that we have these collaborations with them. So we have had one project going on for more than a year, where we have characterized the different effects of different synthetic cannabinoids and their cardiotoxic effects. But the project that I will describe for you here today, that is instead then a description of the effect of different benzodiazepines on their targets to the GABA receptors. So I can just say that these benzodiazepines, or benzos as they are called on the street, they are downers, so you take them to get calm, more or less. So they act on the GABA-A receptor, that is a pentameric ion channel. So when GABA binds, let's see here, so when GABA binds, it will activate and chloride ions will be able to pass through the ion channels. And this will then reduce the excitability of the neuron. So it is a pentameric ion channel that can consist of different alpha, beta, and gamma subunits. And the binding site for GABA is found between a beta and alpha subunit. And then we have these benzodiazepines. So they are positive allosteric modulators of these ion channels. So that means that it still needs GABA to bind, to activate, but they will increase the effect of GABA. And the binding site for the benzodiazepines is instead between an alpha and a gamma subunit. So the first benzodiazepine that was described, that was a drug called Valium, or sold under the brand name Valium. So that was a diazepam. So it was under prescription for short-term use to treat anxiety disorders, insomnia, and also seizures. So it was released or launched in 1963. And they lost their patent in 1985. And since then, there has been a plethora of different versions of the diazepam. But there have also been a lot of other benzodiazepines, also as prescription drugs, and also then later on as designer drugs. So what they all have in common is that they have a benzene ring that is fused to a diazepine ring. And when the compound have these parts, it makes them very addictive. So most of the compounds are very easy to get addicted to. And that is also the reason why they only were prescribed for very short-term use. And unfortunately, they are also quite easy to overdose, some very easy to overdose. And the overdose of these compounds, that can cause extreme drowsiness. It can also cause respiratory depression, coma, and also possibly death. So it's really dangerous compounds when you use them in the wrong way. So benzodiazepines have been found on the illicit market for quite many years now. So the first one to be found, to be detected, that was phenazepam. And phenazepam was actually, or is actually, a prescription drug in Russia that was developed in the 70s, but it was found then on the illicit market during 2007. And we will come back to this compound later on in the presentation. And since then, additionally 35 compounds have been identified on the illicit markets. So it's really a lot of different compounds that's available there. So these compounds, as I said, they are very dangerous when they are used by themselves. But many of the addicts in primarily, they are combining them with other different drugs. So other psychoactive drugs, so like cocaine and cannabis, and also with alcohol. And that is very common to combine these drugs with alcohol. And this increases the effect of the benzos, but it will also dramatically increase the risks with these compounds. So last year, the benzodiazepines, they were detected in about 60% of the deaths that were connected to the different drugs used in Scotland last year. And for many of the compounds that I showed you on the last slide, there's little to no information that is known about their effect on their targets, so on the GABA-A receptor. And that makes it very difficult, first of all, to help the healthcare system when they get the patient with overdoses, how they can help them. But it also makes it more difficult to make these compounds illegal. So it's really needed information by many reasons. So more knowledge is really needed then, both for harm reduction, but also then to be able to classify them as narcotics and making them illegal. So this brings us to the aims with this study. And that is to, first of all, establish an assay that we can study these compounds on, because they are very potent, so it really has to be a good assay for us. And then we also would like to use this assay to evaluate the effect both then of old and also then different novel benzodiazepines that are available today. And in the next step, we would also like to evaluate these compounds then in combinations with other different drugs. So for example, then the alcohol, then to simulate the polydrug use. And to do this, we have used our Q-patch that we have in the lab. So the automated patch clamp technique that uses then the planar patch clamp technique. So for this, we have also used a cell line that we have bought from Visus. So this cell line expresses the GABA receptor as alpha-1, beta-2, gamma-2. And that is one of the most common GABA-A receptors in the brain. So the first step that we did, that was to characterize the GABA, the dose-response curve for the GABA. Also then to finding a concentration that we can have for GABA when we combine it with the different benzodiazepines later on. So first of all, we tested it here, as we can see here then. So we apply GABA, and then it's present for 3 seconds until we wash it out for 3 times. And when we plot dose response, this gives us an EC50 value of approximately 11 micromolar. And we selected a concentration of GABA that corresponded to approximately 10% of the maximum current, because that is kind of a good value to have to be able to detect the effect of different compounds later on. So we chose 2 micromolar of GABA. So then we combined this concentration of GABA with the benzodiazepines. So first, we just made 3 sweeps, so 3 recordings, just in the presence of the GABA to have as a baseline. And then we increased the concentrations of the diazepams. And what we can see here quite easily, that is that we don't reach any steady state current during these 3 seconds when they are co-applied. So this indicates that this is a too short time for these compounds to reach their full effect on the channel. So then we instead pre-treated the cells with these compounds for about 3 minutes. And the first thing that we can notice then, that is that we have steady state currents immediately when we apply the GABA. We have the steady state current, but also that we have an activation at much, much lower concentrations. So here already at 1 nanomolar, we can start to see an activation by the diazepam. So this consequently shifted the EC50 values quite a lot for the assay. So we shifted it from 160 nanomolar that we had without the pre-incubation to down to as low as 28 nanomolar for the diazepam. So the assay that we used then in the coming experiments that I will show you, that was that we used this 2 micromolar GABA, and we used a version of the Q plates that is called the multi-hole Q plates. And before the experiments, we also pre-treated the cells in 30 degrees for approximately 60 to 20 hours, and that was just to promote the expression of the ion channel as well. And we could also see quite early on that DMSO actually had a small blocking effect on this GABA-A receptor. But for us to instead choose ethanol as a dissolvent was not an alternative, since ethanol as such will also activate the GABA channel. So we had to stick with this, but keep the concentrations always the same concentration, and also then as low as possible. And then, as I already told you here, that we use this pre-treatment with the benzos to get the full activation. So we use this assay, first of all, to test a number of prescription benzos that are available. And we can see here then for diazepam, as we already show you here. So then we had an EC50 value of 28. And the EC max value here was 4.2, so we had a 4.2 times increase. And that was actually the highest EC, or increase in the maximum current, so to say. But they are quite similar, all of them. So we can see that the biggest difference is then on the EC50 values. So this is the value that we will focus on later on also for the presentation here. So as I said earlier, phenazepam was one of the first drugs that were identified on the illicit drug market in Europe. And this drug is much more potent than diazepam, for example. So it has an EC50 of as low as 0.9 nanomolar. So consequently, it starts to activate at much lower concentrations than that as well. And the structural difference between these two compounds, that is that the chloride, as we have here on diazepam, is replaced with a bromine. And it's also that we have a chlorine at this position. We will come back to this position later on. And it's also that we have, instead of a methyl group, we just have a hydrogen here. So there are quite some differences between these compounds. So these differences together then increase the potency more than 30 times for these compounds. So then we tested all the other compounds that was available on the market that had been detected on the illicit market. And then we actually plotted their EC50 value versus the year, either when they were approved as prescription drugs or when they were first described. So many of these drugs, they are actually found in the literature. Many of them are found in old patent applications from the drug companies. And then they have been kind of found by the very creative drug designers. So they have used these old compounds as starting points in many ways. So we can see here that many of the designer drugs, they are actually not really new, but they were described much earlier. Then we also have a cluster of the designer drugs that were designed much more recently. And what is quite interesting here also is that this is not like normal drug discovery, because then you normally develop the compounds to become more and more potent. But here, the main aim, that is for them to make these drugs, that they should not be illegal. So that is really what motivates them, how they design these compounds. And there are two compounds that are marked here, and that is alprazolam and flualprazolam. And alprazolam is the drug Xanax that is used a lot in the state, both as prescription drug, but also then on the illicit market. And it is a potent drug as such, but then when you just add a fluorine to this position here, then you reduce the EC50 value by a factor of 3, so you make them much more potent. And this was actually a catastrophe some years ago, because they were sold on the street as real Xanax, but they were fake. So they contained a mix of different benzodiazepines, but a very high concentration of this flualprazolam. And there was actually a lot of death that was caused by this, and also then because these ones were so easy to overdose, because the users, they thought it was alprazolam, so they used it in the same way as they normally do, but then it was instead flualprazolam that had a much higher potency, so it unfortunately killed a lot of people. And then if we focus again on this position here, then we can see that also when we combine it with the halogen on the scaffold of the diazepam, we can also see that it really increases the potency. But interestingly, is that if we instead introduce an halogen at this position, down here, we can see that we turn these compounds from being very potent positive allosteric modulators instead to become an inhibitor of the ion channel. So if we just plot the dose response curves for these two compounds, we can see that we have diclazepam that we have up here, and then we instead have this 4-chlorodiazepam. So just by removing or moving this halogen from this position, and this very super small displacement of the halogen then can have really drastic effects. So what we now are going to do is also to investigate if we can correlate this with existing binding site for these compounds to see if we can understand what's really going on there. So just to summarize, is that we established an assay for these compounds to be tested on the Q-patch. And we also investigated the different effects then over the different years when they were described or when they were prescribed. And then also, I should also mention that the data that they are using quite a lot today to evaluate these different compounds, that is more anecdotal dose response curves, how the user, how they say that they feel that the compound is. Maybe they feel that it's 3 times more potent than alprazolam or something. So this is kind of the data that they have been using quite a lot also for distinguishing them between them. But this really shows that it's very good for them if they also can use the electrophysiology and really then investigate the real effects. So this is a highly ongoing project. So we've just been working with it for a few months. And what we're going to do now is then that we will try to investigate and examine even more than the binding sites for these compounds, because we expect that all of them will bind to the benzo site. But we also think that some of them might bind to an additional site. Because for some of them, we see a really bell-shaped curved dose response curve. And then we think that this could be due to binding to another site that then instead have the opposite effect. And that could actually be very, very, very dangerous, because these patients that take these drugs, if they don't get the full effect, they will take another pill. But if the reason is that they don't see the full effect because they're already on the lower part on the dose response curve, then they could just kind of continue to take them even more and more. And then that becomes super dangerous, of course. So it's very important also whether we're kind of to help the users with these compounds. And then we will also like to use our assay then to examine the activity of the novel benzodiazepines in different mixtures with other drugs, then to kind of to simulate the polydrug use. And Caitlin, who brought this project to us, she was actually a postdoc in Scotland earlier, and then she was characterizing and investigating the use of these different benzodiazepines in Scottish prisons. So this is the reason also why I had some statistics from Scotland. And we also have some seized materials that she have gotten from these prisons. And then we will also try to see that when we investigate these samples, if we can see how the effects are, because they could be a mix of a lot of different things. So with that said, I would like to thank, of course, Henrik and Caitlin for bringing this sometimes very challenging but interesting project to us. And of course, I would like to thank Fredrik and Amaya, for my colleagues at CBCS at LiU, and also Sarah Lean, who's also involved in this project. And then also a super big thanks to Naya, because she has really been a hero for kind of helping us with this assay. So it's been we are super grateful for her help. And you've already seen this picture when Sarah showed you yesterday. So in September, we will organize an ion channel meeting in Linköping. And I would just like to say that you are more than welcome. And you can find information actually on Sophian's website under event. There you can find more information about it and also a link to it. So Linköping is a nice city, and it's a lot of electrophysiologists there. So thank you so much.
Thank you, Nina, for the wonderful presentation. So now, open the question from the floor.
Great talk. But so I'm wondering what is, so now you are doing all these Q-patch experiments, but is there a next step to test also that in manual patch clamp on brain slices or on iPSC-derived neurons to see the effect of those different benzos at different doses?
Yes, I've heard that you will be able to do that on the Q-patch.
Of course we can help.
So, but of course it would be super interesting to investigate, and in particular, as you say, some of these combinations of different drugs. So first, if we do it on the Q-patch, and what we find then, and see how that replicates also then in some other tissues, that would be very interesting. Yeah, absolutely.
But then maybe I have, do you know if benzos, they have also a higher activity on the extrasynaptic GABAergic receptors?
I think that's where they have the main effect, yeah. Okay. Thank you for the questions.
Nice talk, thank you. As you know, I would say pharmacokinetic aspects are also very critical to the addictive potential of these drugs. And I've seen that some of the newer drugs are probably more lipophilic and could cross the blood-brain barrier much quicker. So do you intend to investigate that as well, to get a complete, comprehensive picture?
That's a super good kind of, how to say, option or suggestion, actually. So as you say, just starting to look for them for lipophilicity, how it also kind of, how it correlates with the effect. That is also kind of starting with that point, yeah.
With the, if you can quantify the lipophilicity, you can kind of predict the speed of the crossing of the BBB.
Exactly. And I think them just also kind of the normal ones, I think they are also quite easy to just pass.
Yeah.
I don't even think they see the membrane, I don't know. So.
Yeah. I think that's, for instance, part of the addictive potential of flunitrazepam, which is highly addictive. It's because it crosses very quickly.
Well, thank you. Yeah.
Thank you for beautiful presentation.
Thank you.
And go back to your data. You showed two dose response curves with or without pre-incubation. And I couldn't actually understand the meaning with, without pre-incubation. Well, what did you do?
So what we did was that here, we just apply them immediately together in the first one. So then we only, so the first time that we then apply the GABA, we premix it, the GABA with the compound, and we apply it immediately. And then we wash it out with extracellular solution. What we did here was that we made 3 recordings just with the GABA, as we can see here on the baseline. And then we actually, instead of washing it out with the extracellular solution, we washed out the GABA in this concentration with the next concentration to be tested. So here we actually applied the next concentration to be tested. And then it takes 3 minutes until it goes back. And then we can see that we have reached the steady state current immediately when it activates.
Approximately 3 minutes pre-incubation.
Yes. Yeah.
Okay. And I have another very naive question. Why almost all these benzodiazepines bind to the alpha-gamma boundary of the subunit? Why not other places? Why there?
I think they may bind to other places as well, to be honest. But it says that the benzodiazepines, they have their main effect on the GABA. But it also says that they can also act on some other receptors as well. But I think this is really where they have the high affinity binding site. But I think they will also act on other places, yeah.
Hi. So I have a question. So people usually, people usually take this with alcohol, right? With ethanol. And my question is that, do you think it's to do, so how do you think ethanol actually work with this? Like, does it make membrane more leaky, or is there allosteric site for ethanol to bind and therefore enhance the effect? Or, how does it work together with the ethanol? Because apparently if you get drunk before taking Valium, you become a bit more...
So the thing is that also alcohol acts on GABA. So how these kind of 1 + 1 becomes 3, That is over my knowledge for the moment. But this is, I guess it has to do something also with how they affect the movement then of the receptor when they are there together.
I have a certain naive question.
Thank you.
A recent cryo-EM structure isolated from neurons contained a neurosteroid endogenously. So probably the properties of GABA receptor depends on the cell types. And I think that you record from heterologous expression system. I wonder if such a complex do not include neurosteroid, maybe response could be slightly different. So how can you fill a gap between native channels and heterologous expression channels? For example, have you ever added a neurosteroid to see if the benzodiazepine sensitivity is changed?
No, we have not tested that yet, at least, but I think it's a very good suggestion also then to even more clarify the effects of it. So it's a very good suggestion, so yeah.
Thank you very much.
Thank you.
So just a one short question, last question.
Sorry, just a little bit primitive question. The one slide before this one, when you applied a quite a high concentration GABA,
Yeah.
you can see the little bit of reactivation after washing out. Right
This
in
one
the orange
here?
line. Yeah,
Sorry?
what does it mean?
That is due to the construction of the Q plates.
Okay.
Yeah. So when they constructed the Q plates, they have some hidden corners.
Okay.
So the solution in these ones, it doesn't disappear the first time. So that's also why we always add a solution at least 2 times. But
Thank
it's a
you.
very super observant of you.
Thank you very much.
Yeah, thank you.
Thank you, Nina. And